Hi, good afternoon, everyone. Welcome to today's webinar series, Fighting COVID-19, a webinar series by National Taiwan University and NanoStrings. Before we start today's webinars, a couple of housekeeping notes for the audience. So if you'd like to ask any questions to our speakers, please type your questions in the bottom Q&A sections. Alternative, you can also raise your question in the chat. Today's seminar is the last part of our three-part seminar series, Fighting COVID-19. This event is organized by NanoString and National Taiwan University. In the past 2 weeks, you already had discussed how biomarker discovery can aid in the COVID-19 pandemic, as well as how by utilizing new genomic platforms, we can identify new therapeutics for the pandemic. Today, to conclude this webinar series, we really want to discuss how we can prevent the next pandemic or how to get prepared. As we know, for the North Hemisphere, right now, we already entered the winter seasons, and coronavirus is only getting more serious. So by discussing in this webinar how we can do some prevention for the next pandemic, we hope to conclude this series and bring to shed some light on our next step to fighting the pandemic. And now I would like to introduce the organizers, Prof. Ruby Huang of National Taiwan University, to give us a short opening. Welcome, Ruby.
Thank you, QQ. Thank you, everyone, for joining this webinar. I'm Professor Ruby Huang from National Taiwan University. As the head of international collaboration of NTU Research Center for Epidemic Prevention Science, we organized this webinar series jointly with NanoString to facilitate the cross-border interactions and collaboration, because we believe that the infectious disease has no borders. So the academic collaborations, we should emphasize on local niches with cross-border relevance. So this initiative is supported by Ministry of Science and Technology of Taiwan, aiming to facilitate this kind of international collaboration efforts. So without further ado, I thank you very much for joining this webinar series. Hope you enjoyed today's talk. Back to you, QQ.
Thank you, Prof. Huang. And now I would like to introduce today's speakers before we go into the sessions. So today, our first speaker is with Professor Winni Chen from National Taiwan University. She will discuss about how the receptor-binding domain and how vaccine strategy against COVID-19 can help us to fight this pandemic. Prof. Chen's sections will be followed by Prof. Yes, also from National Taiwan University, who is also going to discuss about antiviral therapeutics in the COVID-19 infections. Last but not least, we had Dr. Kirsty joining us all the way from the University of Queensland in Australia. In this very interesting session, Dr. Kirsty will discuss how what's the relationship between children and the SARS-CoV-19 transmissions. So without further delay, now I would like to pass the presentations to our first speaker, Professor Chen. Hi, Professor Chen. I will stop the sharing and then I will pass the presentation mode to you.
Hi, can you hear me?
Hi, Prof. Chen, I can hear you well and I can see your slides.
Great. All right, thank you for having me today. This is Winni Chen from Department of Veterinary Medicine in National Taiwan University. It is my pleasure to give a presentation about the receptor-binding domain RBD-based vaccine strategies against COVID-19. Here I'm showing you an overall classification for coronaviruses, including animal and human ones. You can find there are 4 different parts, which are alpha coronavirus, beta coronavirus, gamma, and delta. and they all have their respective host. And the ongoing pandemic COVID-19 is caused by the SARS-CoV-2 down here, belonging to the beta coronavirus. But before the emergence of the SARS coronavirus 2, we already have tons of animal and human coronavirus, especially some of the animal coronavirus have been controlled by the vaccines. So when we are developing the countermeasures for the COVID-19, we should consider what we can learn from other pre-existing coronaviruses. This is a typical coronavirus variant on the left, where you can find the spike protein protruding on the outer surface, and the spike protein can be further divided by with two different subunits, which is S1 in the middle and S2. And the receptor binding domain, we call it RBD, is located on the S1 subunit. When doing an infection, RBD will attach to the host receptor on the surface and enter the cells. So the RBD plays the most essential role in establishing the infection. And on your right, among different coronaviruses, the RBD can locate in either N terminus or the C terminus of the S1 protein. But no matter where it is, the RBD is the most critical target for virus prevention. In the past few years, we have been developing the RBD-based vaccines against the MERS coronavirus. We have expressed the viral RBD antigen, which can interact with the DPP4 receptor. And the immunization in mice showed a good neutralizing antibody response comparing to the control group, and significantly reduced the viral load after the virus challenge in the lung. And most importantly, the RBD vaccines protected the mice from the lethal challenge and reached 100% survival. Similarly, we were able to prepare a RBD-based vaccine for chickens to prevent the avian coronavirus, which is called IBV. In this study, we prepared the RBD antigen and allowed that RBD has a good trachea binding specificity. And in the chicken immunization model, by using the subcutaneous immunization, the RBD vaccine induces systemic serum IgG in chicken, or using the intranasal immunization, which is a mucosal immunization, the RBD vaccine also triggered the elevated IgA response in deer. And after virus challenge, the vaccinated chicken were protected and showing alleviated symptoms and the lower virus shedding. So back to COVID-19. There are many types of vaccines currently under development. Today, we will be focusing on these two types of protein subunit vaccine for more discussion. Literally, the RBD, receptor binding domain based, and the full spike protein based vaccines. According to the most updated information, we have nearly 70 types of vaccine candidates that already entered the clinical trials. And for the protein subunit vaccines, the main topic today, there are a couple of them already in the phase 1 on your left, and phase 2 in the middle, and only one in the phase 3 provided by the Novavax company. This is the only one currently in the phase 3 based on the subunit protein. So let's take a look at what the Novavax provided. This company uses a full spike-based protein subunit, including entire S1 and S2 as their vaccine candidate. Basically, the spike protein is engineered and slightly modified to reach a better stability and form a conformational trimer to resemble the native virion. And from the TN image, you can find the spike protein form complex after formulating with the adjuvant in the indicated by the white circle. However, in terms of vaccine antigen, is the largest full spike better or the smaller spike RBD better as a vaccine antigen? So there might not be a standard answer. So let's take a little comparison. In terms of the production, to express the full spike is much more difficult because in terms of the size, the full spike would be over 100 kilodalton. And for the native trimer, it would be hundreds of kilodalton. And mostly the full spike protein is membrane associated, which makes the purification process even more complicated. And the large size protein sometimes aggregate during the production and not easy to separate. In contrast, the spike protein, the spike RBD protein is relatively small and usually is soluble, not aggregating, and not associated to membrane. So it's easier to produce and purify to a mass production. And in terms of the epitopes, of course, the full spike encompass more epitopes for T cell or B cell activation, but that's the cons for the spike RBD. There is only a few epitopes for the RBD. But in terms of the antibody induced, full spike induced both neutralizing and also non-neutralizing antibodies because still there is S1, the other area in S1 or S2, which is not neutralizing. But for the RBD, they mostly induced neutralizing antibodies. In terms of this difference, it caused a little bit difference in the ADE risk. So the full spike inducing possibly non-neutralizing antibodies. So there is moderate to high ADE risk. The ADE refers to the antibody-dependent enhancement. In contrast, spike RBD has a lower risk to induce the ADE. So we recently published a preclinical study using RBD-based vaccine in mice. In this study, we cloned and expressed the RBD of the SARS-CoV-2 in the mammalian cells to harvest the protein antigen in this culture supernatant. After affinity purification, the antigenicity of the RBD are examined using the immunological assays and administered to wild-type mouse for the immunogenicity test. This RBD we have expressed is a 184 amino acid product from the protein modeling on your right. You can find that RBD is a conformational structure and interacts with the ACE indicated in green. And the letter in red indicates the most important binding site called RBM, which is receptor binding motif. So after the RBD protein expressed, we harvest the protein from the culture supernatant, starting at day 2 all the way to the day 5. You can see the protein is abundantly expressed. And using Western blot, we can find the size of RBD is about 25 kilodalton. And the protein yield is pretty decent. We can typically obtain 30 milligram from a liter of the mammalian suspension cell culture. After treatment with the PN-GASE-F, we can find a sharp change of the molecular weight from 25 to 20, indicating our expressed RBD is properly glycosylated. And most importantly, we use the human convalescent serum to validate the antigenicity of our RBD protein. So you can, on your right, you can find that our RBD can be recognized by the human convalescent serum very well. And another important part for the RBD is its bioactivity assay to bind the human ACE2 receptor. From the left, you can find there is a dose-dependent binding for RBD to the human ACE2. And you can see the curve in the middle showing the binding kinetics. So indicating our viral RBD expressed in the mammalian cell system has the great binding bioactivity with the human ACE2. And next, we enter the immunization studies. In addition to our expressed RBD, we also incorporate another commercial RBD protein, which is RBD-FC. That is fused with RBD with the FC portion to enhance its immunogenicity. We use the black 6 mice subcutaneous injection, 2 or 3 doses with RBD-FC or RBD with alone adjuvant. And from the serum response, you can find that after 2 doses of each immunization, you can find pretty good antibody response in the mice. And after the further dose of the RBD, in the blue line, you can find that day 63, they can reach the very high antibody titer. And in terms of the neutralizing response, actually we can find the RBD-FC and RBD, they both reach a decent neutralizing titer in this figure. So for summary for the first part, the mammalian cell produced RBD antigen is capable of binding human ACE2 and can be recognized by human convalescent serum. 3 doses of RBD formulated with the aluminum hydrogel adjuvant elicited neutralizing antibodies against SARS-CoV-2 in wild-type mouse. But we keep thinking what we can do to enhance the RBD's immunogenicity. So here we used our patented platform, which is a polymeric hollow nanoparticle, to incorporate antigen and the adjuvant. So the mammalian expressed RBD is incorporated in the core of the nanoparticle. We also incorporate the soluble adjuvant CpG-ODN 2395 in with the adjuvant in the core. And then these 2 elements can be delivered by the polymeric nanoparticle as a shell carrier. After preparation of the nanoparticle, by judging by the TM image, you can find that these nanoparticles are thin shell, hollow nanoparticle, and the antigen and adjuvant can be co-encapsulated in the core of the nanoparticle. And the release kinetics show us that when encapsulating, when nanoparticulated, the antigen and adjuvant can reach a slow release for a couple of days and the release into the physiological adjuvant, the environment. So we checked the immunization result. The most interesting part, we found that only 1 dose of RBD nanoparticle vaccine can induce potent antibody response. So at day 21 after the immunization, we can find the nanoparticle formulation can reach a pretty high antibody titer. And it's even increasing at day 35. For the Th1 and the Th2 responses, we excitingly found that our nanoparticle vaccine in mice got a very balanced Th1 and the Th2 responses comparing to the conventional RBD alone, which can only induce the Th2 response. And very excitingly, we also found that our RBD nanoparticle vaccine can elicit RBD-specific CD4 T-cell response. So on your left, you can find the RBD alone vaccinated mice. We got the lower than 0.1% CD4 T-cell response. But for the nanoparticle vaccinated mice, we got more than 3% CD4 T cell response. And then in the last, we found the neutralizing titers, after one dose, the nanoparticle received mice can have a very good neutralizing antibody response, whereas the RBD alone, the 1 or 2 dose cannot reach any good neutralizing titer. The last but not least, we all want to make the vaccine work. So the scalability and the thermal stability features for a vaccine is pretty important. So our nanoparticle platform, starting from the microfluidization technology, and the particle will be collected by tangential flow technology. And most importantly, it can be lyophilized to store at room temperature. So through this platform technology, we can prepare the nanoparticle vaccine to a very large batch to supply to very many doses. And for the stability on your right, you can find that the nanoparticle remain very stable at least 2 weeks in the Celsius 4 degree. And after lyophilization, it can be stored at room temperature for a very long time. So overall, our nanoparticle RBD vaccine is biocompatible, biodegradable, because PLGA is FDA approved material. And this particle can be produced to a very high production yield. And it's hollow, versatile to deliver different cargo. And it could be storable by lyophilization. For a summary for this part, 1 dose of nanoparticulate RBD induced potent humoral response with a balanced Th1 and Th2 antibody profile. And nanoparticle delivered RBD triggered CD4 T cell immunity, enhancing B cell maturation differentiation to reach the neutralizing antibody titer. And most importantly, RBD nanoparticle vaccine is of great scalability and the thermal stability and suitable for further development. There's a couple more things I would like to discuss. Recently, in July, there is another group published a RBD vaccine that also support our finding. In this study, they used 3 different animal models, including mice, rabbit, and a non-human primate. They found that 2 to 3 doses of RBD plus alum can reach a decent neutralizing titer in rabbit and non-human primate. And more excitedly, after challenge judged by the lung pathology, they found RBD alum vaccinated animal can obtain a good protective efficacy, which further support our finding in developing the RBD vaccine. And this study, they also emphasized that RBD vaccine had the best neutralization activity compared with vaccines from other domains, for example, ECD, which is the full spike, and the S1 or the S2 domain. Among them, RBD still is the best. So it is pretty much for today. The take-home message is our findings highlight the importance and provide a rationale for the development of an effective COVID-19 vaccine through the induction of protective immunity against the RBD protein. But RBD is a small subunit and poorly immunogenic. So a nanoparticle carrier is suitable for co-delivering the subunit and the adjuvant that can enhance the immunogenicity. I would like to thank my research team in National Taiwan University and also my collaborator in academia, Seneca, Dr. Chen Minghu, and their colleagues. I would like to thank all the audience for your listening, and I will happy to take questions.
Hi, Prof. Chen. Thank you very much for the sharing. Very interesting findings about RBD proteins. And we will, before we move to the next session, I just had one very quick question. You probably have been approached with this question many times. So because we know this receptor binding domain is based on the spike protein. And can you comment on like the recent mutant that has been found widespread in Europe and America, the D614G? Is that going to impact? How do you think it will going to impact the receptor binding domain?
Yes, thank you for the question. For the small RBD, the very focusing neutralizing response can be reached, and there are CD4 T cell response can help the B cell maturation. So I think the single 1 point mutation would not be affecting too much, and I think that the RBD is stable, and there are still multiple neutralizing epitopes on the RBD. So I don't think that even not only the D614 or other mutation should be tolerable in the RBD vaccines.
Thank you very much for the clarification, and now I feel safer, and it's a really good news for everyone who is waiting to get a vaccine. So thank you, Prof. Chen. And now we will move to the next session by Professor Ye, also from National Taiwan University. So I think Prof. Chen, you can stop the sharing, and then Prof.
Okay.
Ye can start her sharing now.
Can you see my slides?
Hi, Prof. Yes, we can see a slide and we can hear you very well.
Okay, so shall I start?
Yes, please.
Okay. Hi, good afternoon, everyone. It's really my great pleasure to talk about the potential antiviral therapies against SARS-CoV-2 infection. So I'm Shiou-Hwei from the Department of Microbiology at National Taiwan University College of Medicine. So the SARS-CoV-2 infection is really a disaster for human beings. It already causes around 60 million people get infected and around 1.5 million people dead. So this pandemic is very difficult to contain, and it will most likely become a coronavirus with sustained infectivity to human. And in addition to the development of vaccine, actually the most important task for control this pandemic outbreak is to develop the effective antivirus. And the molecules targeted by specific antiviral could be either the viral protein or host proteins critical for the viral replication or immune clearance. Among the various viral proteins, which most scientists focused on to develop the antivirus, the spike protein and the viral enzymes, especially the papain-like protease, 3C-like protease, and RDRP, have attracted a lot of attention. Let's see how this protein works in the viral replication cycle. The spike protein in the virion is the key for viral entry. It will bind to the receptor ACE2 and then get fused with the membrane either in an endosome-dependent or independent manner. And the genomic RNA will get entering into the cytoplasm of the cell. And then the protease will contribute to the process of the polyprotein encoded by open reading frame 1A and 1AB, which will generate the non-structural proteins to form the replication complex and critical for viral replication. For RDRP, it is the critical enzyme for viral RNA synthesis. Therefore, the inhibitors targeting to these viral proteins will block the key steps in the viral replication cycle and get the antiviral effect. And more recently, the viral maturation process, including the assembly and the viral release process, become the new target for antiviral therapy. In this slide, you can see an increasing monoclonal antibodies, small peptides, small molecules are increasingly documented to target each of these replication stages. And many of these drugs are derived from repurposing of the existing anti-SARS-CoV or anti-MERS-CoV inhibitors. And many of them have been processed for the clinical trials already. And among the small molecules, the only one approved drug by FDA is the remdesivir, which target to RDRP. And more recently, just released at the November 9th and November 21st, there are 2 monoclonal antibody drugs and released by Elilis and Regeneron, which has been approved by the FDA for the emerging use. However, these 3 drugs actually can be applied to the mild and moderate illness only. And according to the symptom or disease stages and severity, from asymptomatic to critical illness, you can see at the early stage, at mild to moderate illness, the viral replication is the major pathogenic mechanism. However, for the severe illness or critical illness, the pathogenetic mechanism may be due to the inflammation process. Therefore, the antiviral therapy like remdesivir and the antibody therapy like the 2 antibodies, and they can be applied mainly to mild to moderate illness. However, for the severe or critical illness, we need to have some other more effective treatment strategy. That is mainly could be the anti-inflammatory therapy. And in fact, in addition to target the key viral proteins critical for replication, the immune modulators are another category of drug targets for drug development. For example, the interleukin-6 or some other cytokine, pro-inflammatory cytokine like interleukin-8 could be the target for the drug development. As reported in a recent NEJM report, the dexamethasone, which can suppress the inflammation, could could shorten the mortality, decrease the mortality rate, and that's shed great hope for the treatment of severe COVID-19 patients. And another inhibitor to block the interleukin-6 pathway, however, still remain inconclusive and in debate. So in this slide, you can see we update the drug for COVID-19. Actually, in addition to the 3 I just mentioned approved by FDA, some have been discontinued the drug therapies like hydroxychloroquine or the chloroquine and so on. But excitingly, there are increasing lists of the proposed drug therapies, which can be classified by into antivirus, corticosteroids, antiparasitics, and immunotherapy, and some other drugs are actively in the clinical trial. So at this stage, I think that development of more effective antiviral therapy is still a very urgent unmet need. We need to develop it. So in our group, we propose that besides to develop the antiviral via targeting to the specific viral protein, we propose to target the specific host factor instead. the host factors will be critical for the replication of the viral life cycle, which will be the target for us to develop the drug. First of all, we also start to focus on the viral entry step and focused on the fusion step after receptor binding to ACE2. And let's look at this slide that while binding of ACE2 of the spike protein is critical for establish the viral infection, actually proteolytically cleavage of the spike protein by the host protease is critical for the subsequent fusion activity. And such a cleavage by host protease will release the fusion peptide from the spike protein, and which is the key to help fusion of the viral membrane and also the cell membrane. And then the viral genomic RNA will get into the cell. And the first protease to target to cleavage of the spike protein is the TMP-ISS2, which target to the cleavage the site at the S2 domain. And actually, the Dr. Hoffman's team have already reported that if you have inhibitor chemostat to target to inhibit TMP-ISS2, it can block the pseudovirus entry into the cell. And in addition to the TMP-ISS2, actually very interestingly, the genomic analysis has revealed that there is a polybasic stretch of RAR amino acids motif locating at the S1, S2 boundary. And very interestingly, this RAR only exists in the SARS-CoV-2, instead, which is absent in all the other members in the same beta lineage of coronaviruses. So it will be very interesting to ask if such a unique polybasic stress of sequence will be critical for viral replication or not. And we will be very interested to ask if such a cleavage site can be a good target for antiviral development. Aiming to do this, we have conducted an experiment to do the site-directed mutagenesis to mutate this RAR to become ARAR. And such a wild type and mutant spike protein has been expressed in the viral E6 cells. And the immunoplot at the right side, you can see that in the wild type spike expressed cells, the spike can be readily processed into smaller protein fragments, which, however, did not occur in the mutant spike expressed cells. So it just proves that this site indeed it can be cleaved in the cells. And very interestingly, you can notice the result showing the lower panel. In the wild type spike expressed cells, you can see a lot of cell fusion with the cells containing multiple nucleus. However, this synthesion phenotype did not occur in the mutant spike expressed cells. You did not see any synthesion phenotype. So it just raised that this cleavage in the spike protein is critical to mediate the cell cytopathic synthesion phenotype. And such a synthesion phenotype difference can be even more evidently detected by the fluorescence microscope observation. And the middle panel showing the wild type spike expressed cells, you can see a few cells with many nucleus inside the few cells. And this did not occur in the mutant spike expressed cells. And then we would like to further explore if there are any furin inhibitors can inhibit this cleavage. So we have chosen 4 furin inhibitors here. 2 are peptide inhibitors, and the other 2 are small molecule type of inhibitors. And you can see the treatment of these furin inhibitors to the wild-type spike express VRE6 and the CMK and the nefsofluorescein, in brief of NEF, can block the cleavage of spike protein. You will see very little cleavage spike fragment here. Therefore, this result just proves that this cleavage is mediated by the active furin. And again, consistent with the previous spike express cells, we can see that the cells treated with CMK and the NAF will inhibit the syncytium formation, very evidently shown here. So now we have inhibitors can block the cleavage and the mediated syncytium. So we can test the functional importance of this cleavage in the real SARS-CoV-2 virus. So we have infected the SARS-CoV-2 in the viral E6 cells. In the infectious system, we have applied the furin inhibitors in the cell to treat the cells. And you can see here that the virus infection indeed can induce very extensive syncytium formation. That's extensive CPE. And the treatment of CMK and NAPH can block the syncytium formation, also in the virus-infected cells. And we have harvested the soup for the viral titer quantification, which is determined by the standard plug assay. And we also harvest the lysate from the infected cells for the Western blot analysis to see how the viral proteins changed by the treatment. So the result in the right panel, you can see that the treatment of CMK and the NAPH can significantly reduce the viral titer to even more than 99%. So at the same time, you can see the left panel, the immunoblot. You see that not only just the cleave spike protein, but also the full-length spike and also the nucleic capsid protein is significantly decreased, almost undetectable by the CMK and NAPH treatment. So it just suggests that the inhibitor can block the viral replication and decrease the viral titer and also decrease the cytopathic syncytium formation. And such a effect actually is has been demonstrated in a dose-dependent manner. And most importantly, we have tried to check what is the replication step targeted by the inhibitor. So here we have conducted the Western immunoblot and also the Northern blot for the viral RNA in the infected cells. In the cells, you can see that concordant reduction of the protein and the viral RNA in the CMK treat and the NAPH treated cells. It does suggest that the inhibitors targeted to the replication step just upstream of the viral RNA synthesis. And then we would like to further dissect which step that the inhibitors targeted. And so we have tried to treat the cells with the individual inhibitors at different time points during viral infection. And here shows the pre-infection treatment experiment that we have pre-treat the cells with specific inhibitor 1 hour before viral infection. And you can see that the CMK can significantly reduce the viral titer and CPE. And however, NAPH though can also reduce the viral titer and CPE, but to a less significant level. So it suggests that CMK can block the viral entry step and NAPH to a less significant extent. However, in the post-infection treatment experiment, we have very exciting finding that when we treat treat the cells with inhibitors 1 hour post-viral infection, you see that the CMK, the viral titer, is not decreased now. However, the NAPH treatment can significantly, very significantly decrease the viral titer, and also the viral protein and viral RNA become undetectable. So the result just mentioned that the NAPH can also decrease the viral RNA synthesis in addition to the viral entry. Therefore, our study aiming to identify any host factors as the putative target for antiviral development. In fact, we have identified that the protease, the furin family of protease, which can target to cleavage of the spike protein at the S1, S2 boundary furin cleavage site. And such a cleavage is critical for viral syncytium formation and also the viral production. Furthermore, we have identified 2 putative antiviral inhibitors. One is CMK and the other is nefsofluorescein, which can block the viral entry and also block the viral RNA synthesis. And actually, we have also tried to determine the selectivity index of these 2 furin inhibitors. And the selectivity index for CMK is more than 5,000. And the selectivity index for nefsofluorescein is around 6.36. And currently, these 2, especially the nefsofluorescein, has become a lead compound for us to further antiviral drug development. And in fact, we already have some derivatives from nefsofluorescein, which can have SI to reach around 30 to 50. So, okay, I will stop here, and I would like to thank my collaborators in National Taiwan University, College of Medicine, the team led by Professor Shiou-Hwei and Professor P.J. Chen, and their support to make this study feasible. And especially, I would like to thank the major contributors for this study, and Dr. Zheng Yawen, Dr. Zhao Tailing, and Dr. Li Qiaoling for their major contribution to this interesting study. Okay, I will stop here, and thank you all for your listening.
Yeah, Prof. Yeah, thank you very much for the sharing. A very interesting study to looking into the inhibitors as a prophylactic approach for the COVID-19. In the interest of time, we do need to move to the next session, but we welcome all the audience to put in a question in the Q&A, which we can address after the last sessions. So thank you, Prof. Ye, for your sharing again. And now I would like to invite Dr. Kirsty Short from University of Queensland. Dr. Kirsty Short has focused her research on the COVID-19 and also in all the vulnerable groups like children and people with metabolic disease. And just piece of information for all the audience as well is that University of Queensland is the institute that has won several awards regarding the COVID-19 research. So it's a great honor to have Dr. Kirsty joining us today at these sessions. So Dr. Kirsty, I can see your slides well, and please start your presentations. Thank you.
Fantastic. Thank you very much. Can everyone hear me okay?
Yes, we can hear you very well.
Fantastic. Great. So it's my pleasure to be here and to talk to you a little bit about some of the research that we've done looking at the role of children in the transmission of SARS-CoV-2. So the role of children in, sorry, I've got a bit of a lag here. The role of children in SARS-CoV-2 has been very controversial. And at the start of the pandemic, what a lot of people were worried about was that SARS-CoV-2 would be like influenza. Insofar as what we know is that children play a really key role in spreading influenza. And it's for this reason that firstly, we aim to vaccinate children, not necessarily to reduce the disease severity in this age group, but that's more to stop the transmission of the virus in the population. And then we also know during influenza outbreaks that shutting down schools is really, really effective at controlling the spread of the virus in the community. So there was lots of thoughts that maybe SARS-CoV-2 would be the same. But then on the flip side, some of the evidence that emerged very early on in the pandemic was that actually it didn't really seem to be infecting children that much, and children were largely resistant to severe disease. So there was this sort of back and forth, and I think because this topic was about children and we all want children to both be able to have an education and do so in a safe manner, it became a very controversial area. So we wanted to do research into this. So we wanted to look at specifically household transmission clusters of SARS-CoV-2 and say, when you have a case of SARS-CoV-2 in the family, how often is it the child that's bringing that into the family? So how often is the child the index case? And how often is it an adult bringing that into the family? So that's what we did. We looked at household transmission clusters of SARS-CoV-2 across a wide variety of different sources throughout Asia, throughout the US, throughout Europe. And this is essentially what we found. We identified 213, I'm not sure if you can see my pointer, but we identified 213 family clusters of SARS-CoV-2. And of those, only 8 had the child as the index case. So only 8 clusters, or 3% of the clusters, was it the child that brought SARS-CoV-2 into the home. In all the other remaining clusters, it was one of the adults. Now let's compare this to influenza. And here we're just looking at H5N1 because that's something that's been very well documented in terms of household transmission clusters. And in household transmission clusters of H5N1, what we found is that children brought the virus into the family cluster about 55% of the time, so the majority of the time. So that's really highlighting to us that difference between avian influenza or influenza in general and SARS-CoV-2. So we actually looked through this data for a variety of different confounding factors, and we found that this pattern held true no matter if we controlled for the fact that adults are more likely to travel than children, no matter if it controlled for the fact that children may have been in lockdown in terms of going to school. We just consistently saw this trend that children were less likely to be the index case in household transmission clusters. So then we thought, okay, well, if a child is infected in the family, how likely are they to transmit that virus to other members of the family? So we looked at what we call the secondary attack rate if the child was either the index case or if the child, or if an adult was an index case. But unfortunately, we didn't really have enough numbers of children being the index case to determine whether this was statistically different to adults. I will say that we saw a trend towards children being less infectious than adults, but we had really small numbers here. And the only other thing I would add to this is there's just in the last 2 weeks been a study released by the CDC of data in the US, and they actually found the same thing. But again, these numbers are small. So instead what we did is we looked at whether asymptomatic index cases in the family were less infectious than symptomatic index cases. And the reason we did this is because we know that children are more likely to be asymptomatic than adults. And in fact, what we found was that individuals who were infected but asymptomatic were significantly less likely to pass the virus on to family members compared to individuals who were infected but presenting with symptoms. So maybe that's some more evidence suggesting that children are less infectious than adults. But what about on the flip side? What about if you have a child in the family and an adult brings the virus into the family, how likely is the child to be infected? Well, in fact, we have good data where we can compare that. And what we found is that when you look at individuals in the family, children are significantly less likely to be infected with the virus than adults. Now, what's interesting is we don't see a difference between younger children or older children. And this is something that some studies have reported. So it may be that we just don't have the power in our studies to detect this. So just to sum up this body of data, what we suggest is that children are playing a really minor role in SARS-CoV-2 transmission. And this notion is slowly becoming more accepted. Now, this information is really important for two reasons. Firstly, as we know, Europe is in its second round of lockdowns. And what's really interesting is that several countries in Europe have elected to keep schools open despite these lockdown measures. So actually our data would suggest that this is probably a pretty safe approach. And even if a child became infected at school and brought the virus home, they're very unlikely to transmit it to their family members. So that's hopefully a really reassuring message for those people in Europe who are living through these lockdowns. These data also have important implications for vaccination rollout, where we know that the vaccine field is very promising and we suspect that there will be a vaccine available soon. But of course, this isn't available to everybody, and we need to pick priority groups for vaccination. And we would say, based on these data, that children probably shouldn't be a priority group for vaccination, because vaccinated children is not going to stop the spread of this in our community. But even despite all this data, there's still lots of criticism and controversial studies remain, and these are being criticized back and forth for statistical analysis, for incorrect comparisons. So I'm just going to leave that point there and move on to the assumption that why would children be more resistant to infection? Because what the data I've shown you suggests is that if a child is infected, they're exposed to SARS-CoV-2, they're either not infected or it's a very rapid resolution of the infection, so they don't bring it home to the family. And then if you have an adult who's infected in the family, the child is very unlikely to be infected, or they have a very rapid resolution of the infection. So what is it that's so different about children that protects them from this virus? Well, what we wanted to know is if there was something fundamentally different in the antiviral response of nasal epithelial cells from children. Now, the reason that we looked at nasal epithelial cells is because this is the first site in the body that the virus comes in contact with. So this is your body's first chance to fight off this virus. So what we did is we sampled the noses of either adults or a pediatric population, and we cultured these epithelial cells in an air-liquid interface. These epithelial cells then differentiated, and we infected them with SARS-CoV-2. And what we saw was very, very striking. If you look at viral RNA, if you look at viral titers, or if you look via Western blot, you see the exact same trend. Every time SARS-CoV-2 was replicating better in the adult cells than it was in the child cells. And I should acknowledge all of this work was done by a very talented PhD student in my group, Yanshan Zhu, and this is all supported by a collaborator of mine, Kirsty. So we can see in vitro that these child cells, these pediatric cells, have somehow a resistance to SARS-CoV-2 infection. So what is it? Well, one of the things that's been suggested in the literature is that pediatric cells actually express lower levels of the viral receptor ACE2, and as a result, maybe they're not getting infected as much. So we looked at expression of both ACE2 and TMPRSS2 in the pediatric and adult cells. And basically what we found was that there was absolutely no difference. There was nothing that we could find fundamentally different about receptor expression between these young children and these older adults. So instead, we started looking at the host inflammatory response. And when we looked at pro-inflammatory markers, we again didn't really see much of a difference between the cells from children and the cells from adults. But where we did see a really, really big difference is in their interferon response. This is both in terms of the production of interferon lambda, which is one of the key significant interferons that's present at the epithelium, and interferon stimulated genes like ISG15 and the toll-like receptor 8. So what we would suggest with our data is that one of the reasons that children are resistant to infection and not playing a big role in disease transmission is that their nasal epithelium can produce a large amount of interferon that then protects them from viral infection. And I'd just like to acknowledge everyone who contributed to this work, in particular Yanshan, Kirsten, Peter Sly for helping provide the cells, Kenyi Chu for doing a lot of the SARS-CoV-2 work, and of course my lab at the University of Queensland. Thank you very much.
Thank you very much, Kirsty, for the sharing. It's both a relief for us, particularly those who had children, knowing that even the kids, you know, at the kindergartens and the school, there's a less chance that they might get infected or bringing the infections home.
That's
yeah, because
exactly right.
Yes, we had a question. So for for Prof. Ye, so it would be interesting. So will it be possible or interesting to see whether, actually this is a question for both Prof. Ye and Kirsty. And so is it possible to see if the fluorine cleavage functions will be different in kids' cells? Because this finding about the lower infection rate is really confining. I mean, it's something very interesting that we definitely want to explore further.
That would be a very interesting possible collaboration, wouldn't it?
Yes, absolutely. I wonder if Prof. Ye will have something to comment on these questions. So let me have to unmute for Prof. Ye.
Okay. So actually, this phenomena, the effect of furin inhibitor, we have tested in some other cell lines, indeed, and get the same result. Yeah.
Yeah,
Yes.
but there could be something very different about how children, the furin in children works. I don't know, and I don't think that's been shown. But certainly the expression of proteases like TMPRSS2 is not different to adults. So that's why we really think it's all interferon mediated. But it would be interesting to look at activity in children, I agree.
Yeah. So this actually the furin is ubiquitous expressed in many cells. So I think that both children and adults could be susceptible to this furin inhibitor. Yeah.
Yeah, thank you very much, Prof. Ye. And then regarding this very interesting finding about the infection in the children, and maybe I think something that I want to ask Prof. Prof. Chen, that what do you think about the receptor-binding domain vaccine? Would it be possible that the virus will have a different binding efficiency in children's? As I know this might be a very stupid questions because just now Kirsty already shown that the viral role is different for likely the virus will replicate the same way. But I just wonder if Prof. Chen, you will see something different if you had you know, some samples from younger adults or the children.
For this question, I don't think it would be difference between adults and the kids, but I think that vaccine efficiency basically contain difference for different ages. That's why we have formulation for adults and elder people, and something that vaccine may not be suitable for the children under 3 years old, something like that. But that could be a vaccine safety issue. But for the vaccine formulation itself, I don't see any, I wouldn't see any difference between that. Yeah.
Yeah, thank you, Prof. Chen. And we did, this is a very interesting session and we did slightly overrun the sessions, but I think last questions probably to Kirsty regarding the vaccination. So you just now mentioned about that children will be at a lower priority on the vaccination list, given some of the findings. At the beginning, I thought this might be something about a little bit anti-logic because you all want to protect the kids first. But it's interesting to know that actually kids, they are safer in this pandemic. So what do you think, is there any way that we can, you know, keep the kids' infection low from your findings, except from like, even though we don't have to give them a vaccination priority?
Yeah, so I think what we're seeing now is that in Europe, where they have lockdowns, you know, and obviously they have very high rate of community transmission, they're still having children go to school. So there is an inherent level of protection around children. But having said that, if the community transmission goes very high, then children still can get infected. It's just a lower chance of it.
I see.
So what I would say is that when we start seeing vaccinations rolled out, and I certainly know that Australia's policy is that the priority will go to those living, the elderly, in particular those living in care homes and healthcare workers. And then what we'll see is the vaccine being rolled out to individuals who have underlying comorbidities. And then eventually we will get to the rest of the population, including children. So I think the point to note is that children are at very low risk of dying from this infection, and the prioritization for vaccines will be those who are at high risk of dying from the infection, or those who are really at the front line of this battle, and that's the healthcare workers.
Yeah, thank you very much for the clarification. I think it kind of still stopped the grandparents from having their grandchildren for a while, at least.
Yeah, I think that's one of the hardest things of the pandemic for a lot of people.
Absolutely. We need to invent some protective gear that will allow the grandparents to help their children again. So with that, I want to thank all three speakers for their sharings, a really interesting story that give us opinions on how the vaccination in different approach, and also how we can protect the children. And now I want to pass the sharing back to Prof. Huang for her closing remarks regarding this 3-part webinar series.
Thank you everyone for joining our webinar series. I think we have fantastic talk today, from the design of using the RBD in the vaccine, and then now the fantastic, very interesting research on the furin cleavage, and then finally Kirsty talking about the susceptibility of the pediatric populations, and then this will further imply on the policy formulation on how we prioritize on the vaccination, and then even the safe distancing policy. I learned a lot personally, and thank you so much for this opportunity to form, to let us know about your research. I hope that we would have more opportunities for engaging each other for international collaboration. Thank you very much.
Thank you.
Thank you.
Thank you.
And thank you, Prof. Huang, and for other speakers. And again, I also want to thank our audience for joining us on this last part of the webinar series. If you are interested in the previous part of the webinars, you can still view the webinars on demand, even for today's webinars. So with that, I want to thank everybody for the participations. And this is the end of our COVID-19 webinar series. Thank you very much.